Welcome to The Explainer. What if I told you that one of the most powerful tools in all of modern medicine was inspired by, of all people, milkmaids? Today, we're unlocking the science behind vaccination. It's an idea that started simple but has evolved into this incredibly sophisticated shield, basically training our own bodies to fight off invisible enemies.
And this powerful statement right here, it really says it all. It's from Morris Hilleman, a scientist who, you may not know his name, is credited with developing over 40 vaccines. We're talking measles, mumps, meningitis. The guy's work has saved literally millions of lives. So how did we get here? Let's dive in.
Our story kicks off way back in the 18th century, not in some high-tech lab, but with a disease that used to kill more people than cancer or heart disease: smallpox. It was an absolute terror, fatal in up to 30% of cases, and if you survived, you were often scarred for life.
Now, long before we had modern vaccines, there was this thing called variolation. And the idea behind it was actually sound. Introduce a tiny bit of the disease to build up some protection. The problem? It was a massive gamble, because they were using the live, full-strength virus. You were literally playing with fire.
And this is where a physician named Edward Jenner comes into the picture. He made this really critical observation, and then he did something that was totally revolutionary for the time. He ran an experiment. He showed, scientifically, that if you were exposed to a much milder, related virus, it could actually protect you from the really deadly one.
And this is the science behind it, which is just brilliant. Jenner was working with a whole family of large viruses called orthopoxviruses. They all share this double-stranded DNA, or dsDNA, as their genetic blueprint. The smallpox virus is variola, the one from cows is cowpox, and the one that ended up in the global vaccine, vaccinia, is a very close relative. It's this family resemblance that makes the whole thing work, that allows for cross-protection.
Jenner's discovery kicked off this massive global effort that took almost 2 centuries, but it all culminated in this one single monumental year. In 1980, the World Health Assembly declared that smallpox, a disease that had tormented humanity for thousands of years, was completely eradicated. Gone. It was a huge victory for science.
So what is actually happening inside your body when you get a vaccine, right? How does this totally harmless exposure get your system ready to fight off a real invasion? Well, it's all about activating your immune system's special forces, what we call the adaptive immune response.
This process is called active immunization. See, a vaccine introduces a safe piece of a pathogen, and that triggers your B and T lymphocytes. Think of them as your immune system's memory makers and its soldiers to spring into action. They learn to recognize the threat, and most importantly, they create long-lasting memory cells. Now, this is totally different from passive immunization, where you're just given pre-made antibodies, like a baby gets from breast milk or through a treatment like the Nirsevimab monoclonal antibody. That protection is great, but it's immediate and temporary. Okay, let's pop the hood on the modern vaccine toolkit and check out all the different ways scientists have figured out how to train our immune systems. Each one is designed for a very specific purpose. First up, you've got live attenuated vaccines. Now, attenuated is just a fancy word for weakened. Scientists take the real microbe and pass it through cell cultures over and over again, until it basically loses its ability to make us sick, but it still looks enough like the real enemy to get our immune system's attention. This is a classic approach, and it's used for a ton of really well-known viral vaccines, like the ones in the MMR shot for measles, mumps, and rubella, the vaccine for poliovirus, which is a type of enterovirus, and the one for yellow fever. It's also used for bacteria, like the BCG vaccine for tuberculosis, which is actually derived from Mycobacterium bovis, and even an oral vaccine for typhoid fever that uses a weakened strain of Salmonella enterica serovar typhi.
Okay, next tool in the box, killed or inactivated vaccines. This is the way to go when you can't really weaken the microbe safely, or if there's any risk it could change back into its dangerous form. The pathogen is totally dead, but its structure is still there, enough to give your immune system a good look.
These whole, dead microbe vaccines are the foundation for protecting us against a whole range of really serious diseases. We're talking everything from the rabies virus and the Salk polio vaccine to bacteria like Bordetella pertussis, which causes whooping cough, Vibrio cholerae, and even Yersinia pestis, the agent of the plague.
But what if you don't even need the whole microbe? What if you could just show the immune system the most important part, like its most wanted poster? That is the whole idea behind subunit vaccines. They use just the key parts, which makes them super safe and really targeted.
And this can take a few different forms. You could have a toxoid, which is just an inactivated bacterial toxin. Think of the bacteria that cause tetanus or diphtheria, which comes from Corynebacterium diphtheriae. The toxin can't hurt you anymore, but your immune system still reacts to it. Or it can be a single protein, like the hepatitis B surface antigen, which, fun fact, was the very first vaccine ever made using genetic engineering.
Okay, now this is where vaccine science gets really clever. So some bacteria, like Haemophilus influenzae, Neisseria meningitis, and Streptococcus pneumonia, they hide from our immune system inside this sugary capsule. And our bodies are just not good at remembering these sugar molecules. So scientists came up with glycoconjugate vaccines. As you see here, they chemically bolt that weak sugar part onto a strong protein part. This totally tricks the immune system. A B cell grabs the whole package, shows the protein to a powerful helper T cell, and that T cell then supercharges the B cell to make a massive, long-lasting antibody response against that sugary coat. It is a brilliant workaround.
So, now that we've seen all the tools in the toolkit, let's zoom out and look at the huge impact they have, not just on you and me as individuals, but on protecting entire communities. So, to understand community protection, we've got to talk about a term called R0 or R0. It's really just a simple number that tells you how contagious a disease is. An R0 of 2 means that one sick person will, on average, go on to infect 2 other people. Simple as that.
Now, let's put that into perspective. A virus like Ebola has an R0 of about 2. The original SARS-CoV-2 was around 4. And then you have measles. Just look at that. Its R0 can be as high as 18. One person can infect an entire room full of people. It's one of the most infectious viruses we know of.
So, what on earth does it take to stop a virus that contagious? You need this level of vaccine coverage, 95%. When that many people in a population are immunized, the virus just has nowhere to go. It hits firewall after firewall. And this creates a protective shield, what we call herd immunity, that protects the most vulnerable people, like babies who are too young for the vaccine or people with weakened immune systems.
And when you actually sit down and look at the benefits versus the risks, the data is just staggering. With a measles infection, your risk of encephalitis, that's brain swelling, is 1 in 1,000. With the vaccine, it's 1 in 1,000,000. Your risk of pneumonia from measles is 1 in 20. From the vaccine, 0. It's not even a close call.
And the benefits go way beyond just preventing that one disease. Get this, because it's both fascinating and terrifying. Research has shown that a measles infection can actually wipe out a huge chunk of your immune system's memory. We're talking 11 to 73% of your antibody library. It causes what scientists call immune amnesia, and it leaves kids vulnerable to all sorts of other infections that they used to be protected from, sometimes for years. So the vaccine doesn't just prevent measles, it protects your entire immune system.
So from a simple observation about milkmaids all the way to genetically engineered antigens, the science of vaccination has completely transformed our world. And with an ever-expanding toolkit that now includes things like adjuvants to boost immune responses and even monoclonal antibodies, it leaves us with a really exciting question. What previously unvaccinatable diseases might we conquer next?